In a previous video, I discussed amino acids and how they are essentially a central carbon, a hydrogen, a amine, which had the nitrogen in there, and then a carboxyl group. Now, if that doesn't ring true, don't worry, we'll catch you all up on that. What in the heck is this on the screen? NH4, rather than going how we got the NH4, see, this is the fertilizer. This is the ammonia, and then we throw in an extra proton in there and add a positive charge, but this is essentially the nitrates or the ammonia that it gets from the fertilizer. And the soybean cell, okay, here's our little soybean cell trying to draw stuff from the ground, and it picks up this stuff here. And in the magic of what this cell can do, this cytosol is just kind of the part of the cell that's just got the fluids in there. And this soybean cell can actually convert from an inorganic ammonia-based thing to a amino acid. One of them is called glutamate and one of them is called glutamine. Between these two, all the other amino acids can be made by this plant. Not every amino acid, but it says other amino acids, meaning that there's some other stuff in here beside those two. And then we have the glutamate synthase. What the heck is that all about? Well, this has to do with the glutamate synthesis of other amino acids. But the key to this is the amino acids, which eventually get into proteins and into bacteria, those will be the items that actually can pull nitrogen from the air and create the ammonia in the first place. So that is the cycle that we're trying to deal with. So if that seems a little weak, maybe run this video over a couple times to see if it makes sense to you. We might just look at glutamate first, and then we'll look at the glutamine and see if we can figure out how those really are amino acids. We wanted to find out first glutamate. Well, this glutamic acid is essentially the glutamate that we're talking about, just a different naming that we can use. And what makes this a amino acid? Well, let's remember we had the amine part. Here's the amine part, the NH2. That we pulled that off the ammonia or the NH4 plus from that previous diagram. And then here's the carboxylate side of the game. Now, how in the heck, I don't see, these diagrams don't look like there's any carbons on there. Where'd the carbon go? Well, each one of these corners represents a carbon, and if we don't come up with the full 4 bonds, then you just kind of have to assume that there's going to be a hydrogen there to fill in the rest of the picture. That's kind of hard to understand, isn't it? But anyway, I'll walk you through it. There's a carbon here, and it's got a double bond with oxygen. So 2 bonds for oxygen, that one's all taken care of. There's a bond between the carbon here, which fits on the amine there, the NH2, and then there's another bond that comes over here, and there's no carbon on this side, it's just the hydroxide ion. So this tells us it's an amino acid. It turns into the amino acid part when you see all this stuff hanging out here. Okay, well, let's start with this carbon. It has to be 4 bonds. Well, here's 1 bond going up to another carbon, and whoa, where's the 4th one? Well, the 4th one is really a hydrogen that sticks out here on the side here that doesn't show up on the diagram, that keeps it clean. So there's a carbon here, and there has to be an H and an H because you got to have 4 bonds. There's a carbon here, and you got to have, let's see, an H and an H, because you have to have the 4 bonds. There's a carbon here, and this looks very similar to this one over here, doesn't it? But anyway, that is the difference between just the amino acid base, and this is the attachment to it. In all of these pictures, the green is the uniqueness to that particular amino acid. Now let's see if I can find glutamine. It's got to be on here somewhere. Let's find glutamine. There's glutamine right there. So here we find the amine, and the difference is the glutamine has some more nitrogen added to it. It's got another H2N that kind of looks like this thing over here. And it's got a carbon and an oxygen. And right now, it's not important that we try to memorize this structure. The important part is that this is what makes the difference between the amino acids. And the amino acids are all different by combining these in the correct order, according to what the DNA code tells it. We come up with the proteins and all the rest of that.